Thank you very much for the very kind introduction. And as he mentioned, first I'd like to thank the organizers for having me. It's a great pleasure to be here. And I will be talking to you about cloning stem cells in the future of life. The first part of my presentation will focus on the use of cloning and stem cells to resurrect extinct and endangered animals, and also the nature of life itself. And the second part of my presentation will focus on the medical applications, and in particular, the use of stem cells to treat or potentially even cure a range of horrific diseases that have plagued humanity for literally thousands of years. So there are essentially two ways to make copies of cells and organisms. The first is known as, and most controversial of course, is known as cloning, also somatic cell nuclear transfer. That's also abbreviated as SCNT. And what you do here, the concept is very simple. You start here with this egg. This is an empty egg. That's the large circle. And you take the cell you want to clone and place it right next to that empty egg. And then when you send an electrical charge through the unit, what happens is the two cells fuse, and the DNA from the cell you want to clone dumps into that empty egg, and then you add some chemicals and you fool that unit into thinking it was fertilized. It starts to divide and it creates a pre-implantation embryo, perhaps no bigger than a grain of sand. And then you can do one of two things with that. You can plate it in a petri dish and create what's known as embryonic stem cells. And these are the body's master cells. They're immortal, they grow forever, and they can turn into virtually every cell type in the body. The other thing you could do with that is you can place that embryo into a surrogate animal and create an entire organism, such as Dolly the sheep. A newer approach is known as cell reprogramming. In this way, you avoid much of the controversy, the need for eggs or embryos, and you can just take, say, a skin cell or some blood cells, and then you add in certain transcription factors or certain chemicals, and you actually bring those cells backward in time to an embryonic state, very much like an embryonic stem cell. And indeed, those cells can make virtually all the cell types of the body. And more recently, there are techniques now where you can actually use those cells to clone. So let's start with cloning. To date, about 24 different species have been cloned. Back in 1958, John Gordon cloned the first animal. That was a frog. Since that time, of course, there's been Dolly the cloned sheep. We and others have cloned many, many other species, mice, rabbits, goats, and even cats and dogs. In fact, back in 1990s, we actually cloned this herd of cows from some cells that were genetically engineered. In this particular case, these cows you're looking at are actually making human albumin in their milk, medical products in their milk. But the same technique could be used to reconstruct extinct animals a la Jurassic Park. In this case, what we did is we took some skin cells from the ear of the animal, and we knocked in a gene cassette, and then using ordinary cow eggs, we were actually able to create this herd of animals. But if you wanted to, for instance, generate something like a mammoth, you could start with, for example, an elephant cell, knock in the genes, and we now, with our new synthetic biology, have the tools to do that, knock in the genes for tusks, for long hair, or even hemoglobin, so the animal could live in a cold climate. And then using techniques that I'll describe a little bit later, we can actually create sperm and eggs and then create an entire organism. So there are two types of cloning, and if you're thinking about cloning endangered or extinct animals, you need to consider a different type of cloning. So the traditional method is something known as intraspecies cloning, and what you do there is you take a cell from an animal that you want to clone and use the egg from the same species to generate that animal. There's another method known as interspecies, which is also can be called cross-species cloning, where you use the egg from a different species than the animal you want to clone. And that is, of course, important, certainly for an extinct animal, but more importantly, if you want to rescue endangered animals where there are very few females. So back in 2000, we actually used that technique, this cross-species cloning approach, to clone the first endangered animal known as a gaur. This is an Asian ox on the verge of extinction. And when I attempted to do that initially, they said, you know, that was impossible, it couldn't be done. And the reason for that is that a clone really isn't a clone. It turns out that every cell has two genomes. One of those is the mitochondrial genome, and the mitochondria are the organelles in a cell that are responsible for making the energy. And those are actually maternally inherited, which means that they will actually be inherited through the egg. The other genome is the nuclear genome, and that's like the DNA that we have in our chromosomes that distinguish us from, say, a mouse or an elephant. And it turns out that those two genomes have to talk to one another. And they can only do that within about 8 to 18 million years species radiation. That means that the species have to be very closely related, or what we call as concordant xenogenic combinations. So we got around that problem by using these closely related combinations. So for instance, the gaur is in the Bose family, as is an ordinary cow. So we can use those two cells to try to achieve this cross-species cloning. So we did this, and what you're seeing here look like probably just little circles to you, but these are actually beautiful little gaur embryos. Blastocysts is what we call them, little hollow spheres. And it turns out that that little spot you see right here is the inner cell mass, and this outer sphere is the terpectoderm, and that outer sphere will attach to and become the placenta and extraembryonic membranes, and this little circle here will go on to become the organism, and in this case, the gaur. And so what we did here was we were planning to make these embryos and send them to Iowa to be implanted into a group of cows. So we sent them out by FedEx. And in this particular experiment, when I came back the next morning, they were still outside the door. The guy didn't pick them up. So eventually, we got around to repeating this. This time, FedEx did deliver them out to Iowa, and they were implanted into these animals. And we had about a 25% pregnancy rate. We let 2 of those continue on to develop. Unfortunately, one of those aborted very late stage. But the other animal we let continue on. And what you're seeing here is Bessie, and she's pregnant 9 months with what we thought was a gaur. We were very concerned about this because the whole world was following us. CNN was playing this every night, and I was thinking, holy crap, what if this Bessie gives rise to an ordinary cow? And that has happened before. It would be very embarrassing. So it turns out, in fact, that it did give rise to a gaur, a beautiful little Bambi-like creature. And everything we thought was fine. Then 2 days later, the animal died of ordinary dysentery, and everyone said, see, Bob, it doesn't work. So I went back, sort of a little despaired for a couple years, and then eventually thought I'd try it again. This time I approached the San Diego Frozen Zoo, who had a number of specimens from animals, and asked them, well, what would really be of great conservation value to you? And Oliver Ryder, who's the head geneticist, said, hey, well, you know, we have the species survival program for an animal called the banteng, and there's only a couple thousand of those left on the earth, and we are responsible for trying to maintain that diversity, but our herd is very inbred, and we need some new genetics to give those animals, you know, a fighting chance to survive. And it just so happens we have a vial of cells from an animal that had been frozen down for a quarter of a century that the animal never contributed to the gene pool. And, you know, Bob, could you really make us resurrect that animal? And I said, send me the vial of cells, and we'll see if this cross-species cloning works. And so they sent us these cells, and we used some ordinary cow eggs from a slaughterhouse, and did this, sent the little banteng embryos out to Iowa, and indeed, we got a beautiful little banteng. Actually, it was born April 1st in 2003. And then we didn't want the banteng to grow up as an Iowa cow, so we sent it back to San Diego to live with the other bantengs there. And this is another picture of that. So I like to collect fossils. So when you come to my house, the first thing you see is this big brontosaurus femur. It's 6 feet long, it weighs 800 pounds, and everyone says, Bob, are you going to clone it? And, you know, first of all, this animal is like 65 tons bigger than my house, and I don't even know what the surrogate would be. But it's interesting, I live on an island, and a USA reporter once said to me, you know, you already have the island, you just need the electric fence, of course referring to Jurassic Park. And I said to him, you can't clone from stone, so don't expect to see any dinosaurs in your backyard anytime soon. However, that doesn't mean extinction is forever. So what you see here is this is Celia. This is the last Bukato mountain goat that existed on the planet. And in 2000, during a storm, a tree fell and crushed her skull, and she died, and that species went extinct. Now, fortunately, some of the biologists had the foresight, and actually what this picture is of them taking a little skin biopsy and also to put a radio collar on so they could track the animal. And so I took a trip to Zaragoza, Spain, where this animal lives in the Pyrenees Mountains, and talked to the ministers there, and I said, you know, hey, you've got these cells, we can resurrect this species. And they sort of laughed and sort of thought this was all science fiction. And actually, it turns out a few years ago, you know, we and others have cloned goats, that they actually did do this, and we helped send them some protocols, and they actually did take some of those cells and actually had a baby Bucado mountain goat born. Unfortunately, it only lived a few seconds. It died of pulmonary insufficiency. But for a brief time, this species that had been extinct had been resurrected. And I've been in touch with them, and I know that they're planning to repeat this soon. Likewise, there's also this interesting frog here, the gastric brooding frog. This is a project Mike Archer's group in Australia is doing, and they have actually some of these frogs that they had thrown in the freezer, the last ones when they had died, so they still have those cells. And this was an interesting creature. We don't know a lot about it because it went extinct before they could really study it, but this frog would lay its eggs, and right after it laid them, it would eat them, and it turned out that the eggs would actually gestate in the stomach, and these little frogs would be born out their mouth. So I'm part of that project, and so these may be two examples of extinct animals that we may be able to resurrect using this cross-species approach. But it has very severe limitations. There are many, many problems with this type of cloning, as you even know from Dolly the sheep. So we now have before us a new technology. So I had mentioned these iPS cells, this cellular reprogramming. And indeed, Yamanaka just won the Nobel Prize last year for that achievement. And so at the time when he published his paper showing for the first time that he could actually make these human iPS cells in cell, I published a paper in Science mentioning that we could probably also use that exact same technique to rescue genes from endangered species, or indeed to resurrect potentially extinct animals. Let's see here. Okay. So this has actually been done. It turns out that there are a number of tricks that we can actually use. One of them has this fancy name called tetraploid complementation. And the way this works is what you start out with here is a zygote, a fertilized egg, and it starts to divide, and then it gives rise to this blastocyst, this little hollow sphere, and then that eventually implants into the uterus and gives rise to live pups. So the difference here is what you do is you start out with the same animal. Say this is an animal that's white fur color. When it divides to the two-cell stage, you send an electrical charge through the embryo and fuse the two cells so that you have twice the genetic amount of material in that cell, what's called a tetraploid. And it turns out when you do that, that allows the embryo to go on to become the placenta into this trophectoderm, this round circular structure, but it doesn't give rise to this inner cell mass, which eventually gives rise to the organism. So you have the ability then to inject your reprogrammed iPS cells into that hollow sphere. So say you want to clone a black mouse. You inject the black cells into this tetraploid, and it gives rise to the cloned animal. So what you end up here is the black mouse. So you're essentially cloning using this trick. There are other approaches where you can heat and activate the cell, but it basically works. So this has indeed been achieved. We can use this iPS technology, for instance, to clone, as I had mentioned, endangered species. So you could start out with, any animal you want. You just take a piece of skin, and then you throw in some chemicals or some transcription factors, turn those cells into the iPS cells, and it turns out we can now turn those cells into sperm and eggs. So I could take a piece of skin from anyone, whether it's male or female, and then eventually be able to make eggs or sperm from anybody. So this may revolutionize reproductive medicine in the years to come. But in the meantime, for instance, people have actually tested if that will really work. So last year, a group in Japan had actually used this iPS technology, took some skin cells and created eggs that gave rise to live pups. The year before, actually, the same group, again, used this IPS technology and created sperm that gave rise to live pups. So we know it works, and so you can imagine, for instance, if you wanted to go back to the mammoth example, where we could just take the elephant cells, take some skin cells, we add in these various transcription factors, make our IPS cells, and then just turn those into the sperm and the eggs. But what you can do is once you have your skin cells from the elephant in your Petri dish, like we did with the cows that make the albumin in their milk, we can then knock in these genes, and we've now, because of the new genome program, we can now get the sequence for many of these animals, so we can get those fragments of DNA, and we know what those mammoths had and how they differ from elephants, and we can then knock in the genes for tusks along here, and then again, using this approach here, create an entire organism. So again, you have the potential here to make lots of modifications, to life, and of course that in and of itself can raise a whole can of worms. So what I'm going to do now is switch to the use of using cloning and stem cells actually in medicine. So in medicine, in regenerative medicine in particular, there are two major hurdles. One is the shortage of tissue. So as we all know, if you need a kidney or a heart, there just aren't very many of those to go around. And also, you have the problem of immune rejection. You can't just plunk those cells or those organs into the body without rejection. So with the advent of pluripotent stem cells, that is, these embryonic stem cells, the cloning techniques and the IPS techniques, we now have a way to solve both of those problems. So this experiment I carried out a number of years ago illustrates the power of actually this technology. So these, again, are some very old cows equivalent to humans at maybe 70 or 80 years old. And what we did in this case, we took a skin cell, again, from the ear. Now, what we wanted to do was create cloned hematopoietic cells. And it turns out that if you clone a cell, it's the same as the animal you cloned it from. So we had to knock in a gene so that we could discern those cloned cells from the self. So we knocked in this case, a neomycin gene. And we were able to then, once we created these hematopoietic cells using this nuclear transfer procedure, we were able to then inject them back into the animal. Now, normally when you inject bone marrow cells or hematopoietic cells into a person or an animal, those cells, you have to use chemicals, myeloablation, myelosuppression, to knock out part of your bone marrow so there's room for these cells to take up home and thrive. In this case, we didn't need to use those chemicals because it turns out not only IPS cells, but also embryonic stem cells are very youthful. It resets the clock. In fact, we published a paper after Dolly the cloned sheep and showed that not only can you take cells that are completely senescent and old and bring them back and make them youthful, but they actually sometimes live maybe sometimes 2 or 3 times longer than the original cell type could. So in this case, these cells took up home through a process known as competitive repopulation, and were very aggressive, even 5 to 10 times more aggressive and able to repopulate than the normal stem cells in your body. And we looked at the periphery of the blood, and what we saw here was the appearance of the neomycin cassette here. You see it appearing in the first 12 weeks. So indeed, that represents the cloned cells appearing in these animals. And then when at half a year, we actually looked at some of these colony-forming cells that came from the periphery of the blood, and it turns out that 60% of those were from the cloned cells, and they created these giant white blood cell colonies, which would normally not mean much to anybody, but we know that we only see those in very, very young animals. So we knew that these old cows had new immune cells. Now, the goal here wasn't just to give an old cow a new immune system. It turns out that we humans have over 100 different autoimmune diseases that the same process could be used for to treat. Indeed, we could use this, for instance, for even a patient who may have AIDS, where we could knock out, for instance, the receptors that the virus needs to get into the cell and just clone up new cells that would be resistant. But I will talk to you about a number of different diseases that we have actually started to explore using this approach. This is another experiment that we carried out. This is with Piero Anniverso, who's now at Harvard, but this was a myocardial infarct model. This is a mouse model that had, these mice had severe heart attacks. So what you're seeing here is a cross-section of the heart. This is the normal healthy heart tissue, and this is the infarcted tissue, which would be mainly scar tissue. And again, we used the cloning approach. We took some cells, we cloned up, in this case, these cells called c-KIT positive cells, and we injected a small number of these cells at the margin of the infarct, and within 1 day, and then these cells were actually had a label in them called LAC-Z, which is blue. So here you can see that after injection in 1 month, that blue represents the cloned cells migrating through the infarcted tissue, and at 1 month time, we had replaced 38, almost 40% of the infarcted tissue in those animals. And not only did we make new myocardium or new heart tissue, but those cells also created a robust vasculature, which was essential to maintain that tissue. So that shows you the power of cloning, and iPS cells are virtually the same. We can do the same trick. We can take a skin cell from anyone in this room and make you the equivalent of embryonic stem cells, and it can be done very easily in the lab, and there are more and more techniques. And as a matter of fact, there are new techniques that were just announced last week and some other ones the month before that. So the question is, you know, are we ready to use those cells to help people in the clinic? So the advantage of this iPS technology is, first of all, the cells are immortal. They grow forever. So we have a master cell bank, for instance, from our embryonic stem cells that we made from 1 cell. And that's enough to treat virtually everyone on the planet. So they just grow immortally. Secondly is you don't need eggs like you do in cloning, and you don't need to generate any embryos. So it avoids all the controversy. But that being said, Yamanaka, who just won the Nobel Prize, the way he did it was he actually used viruses to carry these genes that he needed upregulated into the animal, or into the cell. And it turns out that those viruses, that's not good for the FDA or the regulatory agencies. You really cannot be using viruses in genetically modifying cells if you want a clinical therapy. So that was the first problem. And two is we published a paper for the first time when there was all this excitement about these cells saying, whoa, hold on, there's some real serious problems. And what we showed is that when we attempted to use this technology to make, for instance, red blood cells or other cell types, that those cells, while they looked like they could do the same tricks, they petered out real quickly. They were prematurely senescent. They had very limited expansion capacity, and and they started to die off and apoptose. So this just gives you an example of what's going on. So if you have embryonic stem cells, and say you want to make red blood cells, for patients, you have to go through this, what's called erythroid expansion. So these are little colonies of cells that you want to multiply and grow, and you can see lots of them growing. And then if you look at doing the same thing here with the iPS cells, you see the clusters are way smaller. It turns out that there was 1,000-fold difference in the ability of the iPS cells to make these cells. And we saw this with the retinal cells we were using to try to treat blindness, that they would senesce early. So there were severe problems. And it turns out what was going on, it actually made sense, is that they're throwing in all these genes using the viruses to change, they're getting thrown into the genome. So they're breaking up the genome in hundreds, if not thousands of places. It's sort of like shooting a shotgun at the genome. And so it's going to mess up the behavior of those cells. So we and others have figured out ways to do this now without using the viruses, without genetic modification. So we published a paper, this is with Dr. Kim's group at Harvard, where we can now just make these special proteins that we can just throw in the Petri dish that'll do the same thing. And there are other methods now, there's episomal methods and microRNA and all sorts of fancy approaches where we now have ways to do that, and we can solve most of those problems that I mentioned to you. So we're now at the point where we've actually had a pre-IND meeting, for instance, with the FDA to try to use platelets for patients. So we're right on the verge of trying to get this into the clinic. However, in the meantime, the first likely applications of stem cells are going to be in what we call immune privileged sites. So it turns out there are special places in the body, such as the brain and the eye, that are what we call immune privileged. That means that they don't undergo rejection as severely as the rest of the body. So you can transplant cells that you make from just a leftover embryo for everybody without worrying about that rejection. So we've decided to focus in on the eye. So we have a number of programs to try to treat different eye diseases. One is the use of what's known as retinal pigment epithelium to treat macular degeneration, which I'll talk a little bit more about. We have just gotten FDA approval now to use those cells for myopia. We also have another program where we're using these ganglioprogenitors to try to treat glaucoma. We have a program where we're making corneal endothelium to treat corneal disease. There are cells known as mesenchymal stem cells that we can also use for glaucoma, uveitis, and a number of other diseases. And also these retinal neuroprogenitors that we can use to hopefully reconnect up new photoreceptors. These are the cones and the rods that we see for people who may already be blind. So first I want to talk to you about our clinical program right now. We received FDA approval to do human clinical trials using embryonic stem cells to make these RPE to treat macular degeneration. We're currently the only group in the world doing clinical trials with pluripotent stem cells, with embryonic stem cells. And we also have trials that were approved in Europe, in the UK. So it turns out that dry age-related macular degeneration is literally the leading cause of blindness in the developed world. There's also an orphan indication called Stargardt's disease, which is the leading form of juvenile blindness. So we have approval to study both of those different eye diseases. And AMD alone affects over 30 million people worldwide. And it turns out that this is due to a degeneration of retinal certain pigmented cells in your eye. And so we think with stem cells now, we have the potential to create an unlimited source of these cells for therapeutic purposes. It turns out that the only source in the past that they could get was from aborted fetuses, and you can imagine trying to get some of these, a tiny group of cells out of the eye of these, it just didn't make any sense at all. So if you look at the eye, this is a cartoon of the eye. The light enters the eye, and it hits these photoreceptors. Again, these are the cones and the rods that we see black and white with in color. And underneath them are these RPE cells. And those RPE cells are the life support to photoreceptors. So they do all sorts of important things, and I couldn't even begin to list them all here. But for instance, they provide critical nutrients. They recycle vitamin A. Importantly, what they do is they keep the environment nice and clean, so these photoreceptors can shed up to 10% of their mass in a day, and that they have to go somewhere. And so these RPE cells eat them. They phagocytose them, and so they keep the environment that's like sweeping up the environment. And so there are other things. They're pigmented, so they help absorb stray light. So there are a number of advantages to this new stem cell technology. So unlike when you have to donate a kidney or a certain bone marrow, you know, that's a very complicated thing. They're very limited by the source. The thing is as I mentioned, both iPS and embryonic stem cells are immortal. So you can get as many cells as you need. You just thaw out cells from your bank, you can expand them and grow up whatever you need. You don't have to ever worry about donors. Two is, what's important is you can actually generate these cells under what we call GMP conditions, and conditions that are very sterile that allow you to actually make these cells so you don't have to worry about all sorts of pathogens for cytomegalovirus, hepatitis, or AIDS when you get donors. So you can make them with no pathogens, and again, you can characterize these to figure out exactly at what stage you want your cells so they work best for the disease you're trying to treat. And we found that, for instance, in our first indication here, you know, we can make these RPE cells really easily. They grow almost like weeds. So this shows you what happens. So if you have some of these undifferentiated embryonic stem cells in a Petri dish, what you will see is almost 100% of the time, eventually, they will start to develop these little pigmented freckles, and they can be removed mechanically. And then what it turns out that what happens is that once they're removed, they start to have these cells grow out, and they lose their pigmented cobblestone morphology and start to differentiate and fill up the dish. And then once they reach confluence, they become pigmented again. And you can go through this differentiation, dedifferentiation again and again and again, and you can get millions and millions of these cells. So you can get large numbers of these cells very easily. And that's important if you want to be treating a disease that, you know, affects tens of millions of people. And importantly, before you can test things in patients, you need to first make sure you know what you have in your Petri dish. So we've characterized these cells, and I mentioned to you that one of the important things that these cells have to do is they have to keep the environment clean. So we can sort of mimic that by just throwing in little latex beads into the Petri dish, and what you see is that they eat them. So these are inside the cell. This is an electron microscopy. And then once we show that they actually do what we want them to do, we can then study their properties. So this is a gene analysis. So what we want to do is show that these pluripotent cells have a down regulation of the genes that are for undifferentiated cells, and that they turn into RPE cells and have an upregulation of markers that are specific for RPE cells. And we can quantitate that using certain techniques such as flow cytometry. And this is great. You can make these cells, they work great in a Petri dish. So can they do anything in an animal? Do they really do anything that can matter for us? So there are a couple animal models that really are the gold standards in this area. One is the RCS rat, the Royal College of Surgeon rat, and they develop macular degeneration that's very similar to humans. And so what we went in and is we used some of our human RPE cells that we made, and we tested them in these animals, and then we could measure the visual acuity of these animals. And so if you look at this, you can actually see here, this is the visual acuity. As you increase the number of cells to a higher and higher dose, you can see that there's a dose escalation, and that by the time you get to these higher doses, you see 100% improvement in visual acuity over the untreated animals. Similarly, there's a mouse that's similar to the Stargardt's disease, and we can show that the injection of these cells prevents the onset of the disease. But what I find more impressive, and certainly as a scientist, is that when you look at the histology of these animals, that is that when you look at the eyes after the animals have died or are terminated, you can actually do a cross-section and look at what's going on in the eye. So in the untreated animals, what you're seeing here, hard to see, is one layer of photoreceptors left. These animals are blind. They're essentially blind. They can't see. However, in the animals that got the injection of these RPE cells, this is that layer. This is the outer nucleated layer, 5 to 7 layers thick. So we had very significant rescue of these photoreceptors. And this is exactly the process that goes on in people who have macular degeneration. So all this data was put together, presented to the FDA. Now the FDA, in your first clinical studies, isn't so much concerned about what you can do to help the patients as it is, well, first to show that your technology is safe, that you're not going to do any damage. And it turns out that embryonic stem cells, by definition, when you inject them into the body, they have to form tumors. They have to form these things known as teratomas, and they have to turn into all sorts of things. You can actually look at these teratomas, and they will turn in, and they will have hair and bone, and some of them even have whole jaws. So they turn into everything. And of course, when you put something into the eye and you want it to be a retinal cell, you don't want a tooth. So it was very important to the FDA that we don't have these undifferentiated cells. So what we had to do was develop a method to show them that wasn't the case. So we developed an assay where we could detect even one of these undifferentiated cells in over 1 million cells. So if we had 8 million cells, we could throw in 5 and find all of them. And it turns out that actually for our therapy, the highest dose was 200,000 cells. So we could actually discern even if we had 1 cell. But more importantly, even if you throw in 10% of these undifferentiated cells at the end of the manufacturing process, you still don't end up with any. So that was good. We could say that, you know, we can look at these cells in the dish and know that they should be safe. But the FDA says, well, that's not enough. You got to go into animals for their lifetime, and you need to show us that, you know, your calculations and everything are right, and that you don't get these teratomas, because that's what their main concern was. And indeed, we went in and we did that in these animals, and not only that, but they wanted us to actually spike in large amounts of these undifferentiated cells to see, even if we were way off, like 6 orders of magnitude, will we get a teratoma. And indeed, we didn't see any tumors in any of our animals. We studied, you know, hundreds of these animals. Not only did we not see any tumors, but we didn't see any ectopic tissue. That means we didn't see any other cell type other than the cell type we put in there. So based on that, the FDA gave us approval to do the first studies to treat macular degeneration using this technology. We have 2 ongoing trials, again, one for dry AMD and the other one for Stargardt's. We have 4 clinical sites One is Mass Eye and Ear in Massachusetts. The other is Bascom Palmer in Florida, Wills Eye Institute in Philadelphia, and Jules Stein at UCLA out in LA. So we've studied, to date, we've treated 12 patients with dry AMD. They have various amounts of the cells. We've treated 9 patients with Stargardt's disease, and we've treated 9 patients in our European trial, which is being carried out at Mooresfield Eye Hospital. And this just shows you the design of the clinical trial. Each of these clinical trials is 12 patients, so 12 Stargardt's, 12 AMD in the US, and then also 12 Stargardt's in Europe. So we've treated 30 patients to date. And we also recently got approval based on the safety to actually move into patients that have better vision. And the goal of this technology isn't really to reverse blindness. It's that what happens is that when the patients start to lose their retinal cells, when they lose their retinal cells, you no longer are maintaining the healthier photoreceptors. So our goal is to get into patients before they become blind, before they lose the vision, and to put these new healthy RPE in to prevent that from occurring altogether. So we've just now started to go back into these younger patients or patients that have better vision to see whether we can halt that process. But the first patients were mainly to see whether or not it was safe, and these were advanced patients who already were legally blind. And so the trial design was that you start with a low dose of cells. We started with 50,000 cells. We treated 1 patient, and then after we showed it was safe, we wait 6 weeks. It goes to an independent data safety monitoring board, and